recording. I'm going to do attendance. Bryant is here, WVSU here. Curry's online. Darby, not here. Fisher, not here. Hamilton is online. Hellman, not here yet. Darby is here and WVSU here. Ashley's here and State here. Crystal's on. Taylor's here. Norris is here. Wearing State stuff. Rollins is not here. Saunders is online. Chamblin is here wearing State gear. Vargas is here. And that should count for everybody. No, Fisher's online. Speaking of online stuff, so I was able to confirm earlier the audio was working. So I'm going to confirm one more time that audio is working for me online.
It's working.
Thank you. All right, good. That should help. So anyway, let's do announcements. These will be important announcements. Some of you already saw the one I posted on Facebook, so I'm still sick. I shouldn't be here. Not for me, but for you guys. It's not safe. But yeah, I'm still sick. So office hours are going to be online today. Lab will be online. There's, I think, 2 of you have already done the lab, and I sent you a text, or I sent you in the grading. I said specifically said you don't have to come to lab. So for you guys, you don't need to worry about this. For the rest of you, just meet me online at 10, like you normally would. So I'm not going to be here. We're going to be online. Does that make sense? Same link we usually use. If you don't know the link, email me sometime before then, and I'll send you the link. And we'll meet here at 10, or meet online at 10 a.m. to do the lab. Any questions about lab? All right, exam. This is the last chapter. We're about to start the last chapter before the third exam. So the third exam is all about DNA stuff, right? Whether it be about what DNA does through mitosis and meiosis, or what DNA does during, when we look at genetics, or stuff we're about to talk about now, which is how DNA duplicates and how DNA provides the instructions for proteins. So this will be the end of it. That means probably the exam will be sometime next week. The study guides are out there. I highly recommend doing them. I will say this too, and this hasn't happened with you guys off the top of my head, but I get this every semester almost, except for this semester with you guys. People always tell me, "Oh, I'm not good at testing, or I'm not good at biology, or I'm not good at science," but they don't do the study guides. And I use the study guides to write the exam. So if you guys don't want to do the study guides, that's up to you. Don't lie to yourself and say you're not good at testing, you're not good at biology, if you aren't doing the bare minimum to prepare for it. So look at those study guides if you get a chance. After you've done them, what you could do is meet with me, and I can go through all those questions and say, this is why this one's wrong, or this one's why this is right, or this is how I might reword this one to be on the exam, or this one definitely will not be on the exam, right? I'll go through all those questions with you and help prepare you for the exam. Yes, that's it for exams. I guess that's it. Office hours, labs, exams. Yeah, that's it. No other announcements. Does anybody have any questions? All right. So here we go. The last chapter of cellular stuff. After this, we start dealing with organisms and populations. And to me, in my opinion, I've already said this before, that's when it starts really getting interesting to me. I mean, everyone's got their own stuff that they're interested in, but for me, that's the more interesting stuff. But as far as cellular stuff goes, to me, this is the most interesting. Obviously, because this is what I did my thesis work on, was this kind of stuff. But anyway, let's talk about it. We're jumping into molecular biology. Now, once again, I've decided to stick with the PowerPoint that I used from the previous textbook, and I've just made some slight changes to it. But generally speaking, I'm going to teach it in the same order from the last textbook. It'll be the same information mostly. You'll know when it's not the same information. There'll be some additional information. I'll tell you when it's additional and you don't need to know it. But yeah, it'll be in a slightly different order, but that's okay. For example, this stuff, right? This is from your old textbook. It's not from your new textbook, but I like that your old textbook used to start with these pictures and kind of have like a little guessing game, like why this chapter matters to you. So obviously this guy is holding some vials of something. Any idea? And this is it's shot in the dark, but any guesses of what your the old textbook might be getting at with some dude looking at a vial of some kind of liquid? Oh man, it's a shot in the dark. I'm trying to refresh myself. It says, all life on Earth shares a universal genetic code, so your DNA can be used to genetically modify bacteria and vice versa. So this is probably a picture of a guy working with insulin. So that's where insulin comes from these days, right? We take a human gene, right, that's not found in bacteria, the gene that says, hey, produce insulin. We cut it out of the human genome, we put it into bacteria because bacteria are so great at reproducing and easy to maintain, right? So then they have that human gene in them, but because the universal code of DNA and RNA and all that, all the stuff you're learning about in this chapter, because it's universal, the bacteria doesn't know any better. So it produces human insulin. We're going to talk a little bit about stuff like that in this chapter. Also, that's something you can look up for independent work. Like, it's basically GMOs is what it is. Like when you take the genes from one thing and put them in the other. And there's a lot of interesting stuff out there, I think. Anyway, how about this? You guys can tell what this is. Again, a shot in the dark. Any guesses what your book might be, or what the old textbook might be getting at in the context of molecular biology, which is all about like looking at how we go from DNA to RNA to proteins? Any guesses? That's right. There's something called mad cow disease. We talked about it in the previous chapter when we talked about different diseases and misfolded proteins. So we'll talk about that a little bit again on this chapter. And then finally, you should recognize this. We've talked about it at least once, even though I told you you didn't have to know it yet, and you don't technically have to know it in this chapter either. But what are these things right here? Don't be afraid to have a wrong answer. Surely one of you knows. You just got to be proud. For some reason, you just don't want to speak up. How about this? What is this thing right here? Good. It's a red blood cell. How about this one? I'll give you a hint. And here's something you should know. I guess not necessarily. I said you didn't need to memorize this, but how about pleiotropy? So now does it ring a bell? Because this was the example we used when I explained pleiotropy, which is when you have one gene that affects many characters. Anyway, that's a sickle cell. So in the context of the last chapter, we were saying if you have one gene that's messed up, it changes the shape of one protein, and messing up that one protein changes the shape of the whole cell, which then gives you all these different characters or phenotypes. So that was the example in the last chapter. In this chapter, it says one typo of the DNA can result in sickle cell disease, and that's true. So when I said, again, last chapter, it's one gene that caused this, and I'm going to get more specific, and later in this chapter, you'll learn about it. Not only is it one gene, it's one letter. There was only one letter that was messed up. So we messed up one letter, and that one letter had a huge change in that one protein. And that one protein had a huge change in the whole cell, and now this person has a whole disease. All because there was one letter changed in their DNA. Anyway, any questions about that? All right, moving forward. Again, this is from your old textbook. Your old textbook always started off with this recent story called Biology and Society. So it's like, hey, here's a recent story from the news that has to do, hopefully, with, or sort of with your everyday life, but definitely with this chapter. This was published right before COVID-19. So I assume the next edition of that textbook, instead of talking about Zika, we'll probably talk about COVID-19. But you can read about that if you want. There won't be any questions about this on the study or on the exam or the study guide. But yeah, basically what your book is getting at here is like, hey, you ever heard of this virus? Because it was recently in the news. Well, in this chapter, you're going to learn how viruses work, which is really important, right? So for example, by the end of this chapter, I mean, you're not going to be any kind of expert on viruses, much less the COVID-19 virus, but you're going to have a good basic understanding of them. So if for some reason you wanted to look into the COVID-19 virus or whatever the next pandemic is going to be when it rolls around, then you'll have a good foundation to start with. Now, you won't be starting from scratch. And man, that was the most irritating time in my life, was being online and social media when COVID-19 first hit and saw all the idiots who knew nothing. I'm sorry, I shouldn't say it like that. All the people who had absolutely no idea about how viruses or DNA or RNA and protein work. But the only reason I said idiots, because again, it's okay not to know this stuff, but they're out there like pretending or maybe even thinking that they know this stuff. Like, I'll tell you this about the COVID-19 vaccine, and I'll tell you this about the COVID-19 protein. But no, that's not even close to being correct. So that's what irritates me. I don't mind people not knowing stuff. Obviously, that's why I'm here to teach you stuff. But when people don't know stuff but pretend they do. Anyway, here's how this chapter is going to work. And this is actually very similar to, again, this is from your old textbook. This is actually very similar to the way your new textbook works. First, we're going to talk about DNA structure and replication. The DNA structure part is actually going to be a review, because we already learned about this in chapter 3, I believe it was, when we learned about biological molecules. Remember, we learned about carbohydrates and lipids and proteins. Then we learned about nucleic acids. Well, we're going to learn about it again. So this will be a review. Then we're going to talk about how we actually get from DNA to RNA to protein. Because remember, in the last chapter and in this last lab, we said, what is a gene? And I said a gene is instructions on how to build a protein, right? So we're going to learn how that works in this chapter. And then we're going to talk about viruses and other non-cellular infectious agents. Now, your old textbook kind of told it as a story, so I'm going to tell it too. Not that you need to know these details, but it's sort of like gives us the history. Like, here's what we used to know, and it shows you how we built our knowledge of DNA, which I like. But again, I'm not going to ask you these questions. This isn't a history class, so you don't need to know these dates. But by the late 1800s, scientists knew that DNA was a part of the cells. That's it. That's all they knew. They didn't know what it was made of. They didn't know the structure of it. They certainly didn't know what it did. They didn't know how it worked, like how it replicated, none of that, right? But they did know that there was this stuff, DNA, in the cells in the late 1800s. Then in the 30s, 1930s, someone figured out that there was one kind of molecule that was the basis of inheritance. So remember, last chapter we talked about Mendel, right? And that was 1800s. And like I said, he wasn't even famous until after his death. And this is a good example of that, right? It wasn't until the 1930s that someone said they kind of resurrected his work. It's like, oh my goodness, his patterns of inheritance stuff makes sense. And not only that, but at this point in the 30s, they put it together and say, all right, there is a molecule responsible for all these things that Mendel had figured out. They didn't know what molecule it was, but they knew there was a molecule responsible for it. You can look any of this up for independent work. It's slightly interesting in my book, in my opinion. Because there's hypotheses, obviously. It's not like in the 30s they just said, oh, there's a molecule involved. We don't know, we don't care. No, there were some people who claimed there was this molecule, people claimed it was that molecule, and they were arguing about it. Anyway, a decade later in the 40s, they then discovered, and again, this is kind of separate, they hadn't put all this stuff together yet. In the 40s, they discovered that chromosomes are made of 2 components. We already learned about this in previous chapters, but chromosomes are made up of DNA and protein, like you already know, right? The DNA wraps around the histones, and when that happens, you have things called nucleosomes. Excuse me. So you already know that from previous chapters, and they figured that out in the 40s, and they still hadn't put it all together yet. Then in the 50s, someone figured out that DNA, finally, they figured out that DNA was the molecule that acts as the heredity material, right? So they put all that stuff together that all these other people figured out, and finally they figured out this is it. DNA is the hereditary material. And that then ushered in the era of molecular biology. Your book defines molecular biology as the study of heredity at the molecular level, right? So we've already had a study of heredity, which is genetics. But molecular biology is more looking at it as if you were looking at it in a microscope, right? Instead of just looking at that flower mated with this flower, and these were the offspring. In this case, it's almost like looking into a cell. Like, all right, here's this protein connecting with this DNA molecule, and this creating this molecule, right? You're looking at it in the cellular level. The first word for attendance, I'm going to point to it. I'm not going to say it. So if you're online at home and you've got your eyes closed and you're relaxed, that's fine. Just open your eyes for a second. If you're watching the video, so not if you're live online, but if you're watching the video, instead of just sending me the word, put it in a sentence and answer a question. What do you plan to study this weekend? That's it. So again, send me the word if you're watching the video, not the live stream. If you're watching this video later in the day, send me the word in a sentence and say, tell me whether or not you plan to study this weekend. Speaking of which, is anybody doing anything fun this weekend? No? I plan taking my kids to grits. I think that's fun. That's if I'm not still sick. All right, after that, after they put all that stuff together, came the effort to figure out the DNA structure, right? So they figured out DNA was the material that's responsible for all that stuff I talked to you about in the last chapter, right? All those numbers that Mendel figured out. So then they're trying to figure out how do we figure out the structure? Because So talking about the first bullet point here, like I told you way back in the beginning of the semester, there's a real relationship between structure and function, right? So they knew if they could figure out the structure of DNA, then they could figure out how it works, right? Specifically, we could figure out how it has these unique properties, which are the capacity to store genetic information, right? So every cell stores genetic information. The analogy I used in the last chapter was you can think of your chromosomes as books, and in those books are instructions on how to build proteins, right? So that's storing the genetic information. Then they also have the capacity to copy it. So again, using the same example, if those books that stores all those information to build proteins, imagine those were actually books that stored information on how to build things on campus, like how to build a bench, how to build a building, whatever it may be. And imagine those books, big, heavy old books, the original copies, those were kept in the library. Now, if somebody comes and needs to build a bench next to Farrell Hall, they're not going to come and take the whole book, right? Because that's cumbersome. Plus, they only need a little bit of that information. They don't need the whole book. Plus, it's on the copy, so you don't want to take it out and get it ruined in the weather and the construction workers, right? So you're going to copy it. Same thing here. Cells do the same thing. They have to copy that. They don't bring out the whole book every time you need to build a protein. And then finally, they had to figure out how, again, one of the unique properties is how DNA is passed from one generation to the next. So they didn't know any of this. They knew DNA did all these things, but they didn't know how. And again, the reason they didn't know how was because they didn't know 3D arrangement, right? They didn't know the structure. I'm gonna make sure we're still recording since this is, we've been having issues. Yep, still recording. Amazing. Sorry for the pause, guys. So speaking of which, let's talk about the DNA and RNA structure. And again, this is going to be largely a review from what you've had before. There shouldn't be much new information. You need to know that DNA and RNA are polymers, right? You already knew that. The polymers are called nucleotides. The polymers are called polynucleotides, and the monomers are called nucleotides, right? You already know that. So these nucleic acids, which is what the NA stands for in DNA and RNA, they're made up of a bunch of repeating monomers, and those monomers are called nucleotides. If we're talking about DNA, ready to go. And again, you need to know this. Everything on this slide is important. You do need to know this. There will be some other stuff you don't need to know later, but right now you need to know this. If we're talking about DNA, those are the 4 bases, the 4 nucleotides, and I guess there's no need to actually write this down yet if you don't want to, because we're actually going to talk about the different bases in detail later. And then in RNA, it's the same thing, except, as we're going to talk about later, there is no T in RNA, there's a U instead. So RNA's 4 nucleotides are A, C, U, and G. And you will need to know that. But again, we'll talk about it again later. Here's some pictures. Well, are there any questions about this before we dive into it? Excuse me. Here's some pictures. This picture's not from your current textbook. It's from your old textbook. So if you're reading your old textbook and you don't like the picture in that one and it's not making much sense, then come back to the PowerPoint and take a look. But here's a good drawing of, there's different ways to draw. These are all 3 drawings of DNA. Right? Here's probably the more familiar drawing of the double helix if you're far back there. But if you were to see, there's A's and T's here connected, and C's and G's, A's and T's, T's and A's, G and C, so on and so forth. And this shows the fact there's the actual nucleotide, right? So there's a monomer. There's one nucleotide, there's another, there's another, there's another, there's another, right? So you put a bunch of monomers together to build a polymer. In this case, put a bunch of nucleotides together to build a polynucleotide. And here's the same thing. This is showing you the same exact thing as this, just a different way of showing it. This one's a little bit more easy to understand. It's more basic. This one's a little bit more realistic. Not even that realistic. But it shows you again. You've got all these different parts to the nucleotide that we're going to talk about as we move forward. Excuse me. Anyway, are there any questions about this? All right. So, like I said, these are polynucleotides, right? Or excuse me. That's a monomer. 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 Right? They're all a bunch of monomers that are brought together to form a polymer. in that case, nucleotides to make polynucleotides. And this is known as a sugar phosphate backbone. You need to know this. These nucleotides are joined together by covalent bonds. Now, you don't necessarily need to know this. I mean, it's not going to be a question, but technically it's the sugar of one nucleotide that's bonded to the phosphate of the other. I say you don't need to know that, but later I am going to teach you this, that the nitrogenous base kind of hangs off and is not involved in that process. It's involved in a different process, and you do need to know that, and we'll cross that bridge when we get there. So in a way, by process of elimination, the fact that you need to know this part, one of the three parts does something else, and you need to know what it does, then by definition, yes, technically, then you should know it's the sugar of one phosphate that's connected to the phosphate. Sugar of one nucleotide connected to the phosphate of the other. Again, that won't be a test question. But you do need to know what a sugar phosphate backbone is. That's what we're referring to. It's because it's a sugar bonded to a phosphate. See? Sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. But now one thing I will not do on the exam is show you a picture of that, for example, and say, which one's the sugar? And which one's the phosphate? I'm not going to do that. But you do need to know what the sugar phosphate backbone is. And here's something else that's very important, and it's going to be more important once we get there. But like I said, it's these are covalent bonds that are connecting, right? So the sugar to this phosphate, that's a covalent bond. That's a covalent bond. That's a covalent bond. Right? Now, do you remember when we talked about chemistry, I said there was 3 different types of bonds we talked about: covalent, ionic, and hydrogen. One of them was the strongest, one of them was the weakest, and the other one was obviously just somewhere in the middle. Do you remember what covalent bonds were? Were they strongest, were they the weakest, or were they just somewhere in the middle? Because this is not trivial. This will be important later in this chapter. Any guesses? Any guesses? I told you would have to remember this during the chemistry chapter. I told you it would come back to us. Which one was the strongest, weakest, or in between? It's the strongest, and that one, again, we'll come back to that. So here's something else you need to know that I've already taught you. So this is a review. Nucleotides are made up of 3 parts. First, memorize that fact. Then memorize what 3 parts there are. Of course, we've already talked about it because I just told you. Sugar is bonded to the phosphate, and the nitrogen is not doing anything, or the nitrogenous base isn't doing anything. But here they are again in writing. You need to know this. 3 parts of a nucleotide are nitrogenous base, sugar, and phosphate group. The next word for attendance is sugar. If you're watching the video later in the day, not the live stream, not if you're live right now, but if you're watching the video later, don't just send me the word sugar, put it in a sentence, and tell me your favorite thing to eat that has sugar in it. So I guess you could use the word sugary if you want. My favorite sugary thing to eat is actually this simple syrup and margaritas. So technically I drink it, whatever it may be, whatever. Just use it in a sentence. Don't send me the word, use it in a sentence. Anyway, let's talk about this. So again, memorize that first. Now let me teach you a little bit about it. Let's talk about the nitrogenous base first. Looking at all this, the only really thing that's important for this is this part right here, this last bullet point. There are 4 different versions of the nitrogenous base. You need to know that. And I've already told you that, not just in that last chapter, but I've already told you that one before we even started in this chapter. I told you there's A's, C's, T's, and G's. Technically, I said there's 4 nucleotides, A's, T's, C's, and G's. Now I'm going to get more specific and tell you this is the part that defines what nucleotide, right? It's the nitrogenous base that tells us if it's an A or a T or a C or a G. Because if we're dealing with our DNA, the sugar and the phosphate of all the nucleotides, they're exactly the same. For RNA, the sugar and the phosphate are exactly the same. So the nitrogenous base is the only thing that varies. For anybody who remembers this, it was a while ago, so kudos to you if you do. Remember when we were talking about proteins, the monomer of a protein is an amino acid. Remember, amino acids have 4 parts. There's the carbon, the central carbon, a hydrogen. But the important part of all that was the side group, right? The amino acid was defined by its side group because everything else was the same on any given amino acid. They were all the same except that one part. So that one part defined the amino acid. Same here. It is the nitrogenous base that defines the nucleotide. Yes, it does project from the backbone. That won't necessarily be a test question, but that will be slightly important later when we talk about, once we figure out the structure of the DNA, or once they figured out the structure of the DNA, then they figured out the function, how it worked, and that would be important at that point. But again, no test questions. Another thing you definitely don't need to know is that they have rings of nitrogen and carbon. So here's, again, that's the nitrogenous base right there. That one happens to be T, or this nucleotide happens to be T. But yeah, you can see there's a nitrogen here, and a bunch of carbons, right? That's what the rings are made up of. You don't need to know that, but there you go. Anyway, any questions about the nitrogenous base? All right, next is the sugar. You don't need to know any of this. As far as I'm concerned, all you need to know is that sugar is one of the 3 parts of the nucleotide. But your book goes into detail, so I'm going to briefly discuss it. So that way when you're reading it, at least you'll know what I'm talking about. And hopefully you'll remember, oh yeah, he said I don't need to know this. But here you go. The sugar, if we're talking about either way, the sugar has 5 carbon atoms. You can see it right here. 1, 2, 3, 4, 5. That's the 5 carbon atoms. 4 of them, again, as you can see right there, are in the ring. One of them is above the ring. So there's the one that's above the ring. It looks like it's not part of the sugar, but it technically is. The ring always includes an oxygen atom. You can see this right here. At least when we're talking about DNA, right? And the carbons are numbered clockwise away from the oxygen. So clockwise goes like this, right? Here's the oxygen, right? So here's the first carbon. So that's what we call 1', 2', 3', 4', and 5'. But you really don't need to know that. I do think it's important at least to bring this one up because anytime you read anything good about DNA, at least the molecular structure, you're going to be reading these things, right? So hopefully as you're studying and you're looking at other sources other than this textbook and you're reading about DNA polymerase and RNA polymerase, all these things you're going to learn about later, you're going to see these words like 1', 2', 3'. So I don't want it to confuse you. If you're really into it, or if you're one of those students, you need to know exactly what it means. Now you do. But what I'm telling you is you don't need to know. So as you're hearing these things, again, if you're studying and you're reading this, like a 1', it goes from 1' to 5'. What does that mean? Right? So just know you don't need to know that detail. Anyway, any questions about the sugar? Excuse me. All right. The phosphate group is similar to the sugar academically in that all you need to know is there's a phosphate group. So your book included this other information. I'm not going to ask you. Again, since your book included it, I'm going to talk about it very quickly. Obviously, there's a phosphate in it, hence the name phosphate group. It just happens to be at the center. Another thing, just so you know, this is where it gets its term acid, right? So these are nucleic acids. Deoxyribonucleic acid. Ribonucleic acid, that's where the acid comes from. But I'm not going to ask you that. And also it happens to have a negative charge on one of the oxygen atoms. I'm not going to ask you that either. That was important later. That was important earlier when we talked about ATP. Bless you. Because remember I was telling you ATP is a really highly energetic molecule because it took a lot of energy to put those phosphate groups together because they're so negative. So that was important back then. It's not important anymore. So again, know the 3 parts of the nucleotides: nitrogenous base, sugar, and phosphate group. Also know that you can shorthand that. For the exam, I'm not going to try to trick you up. So I'm not going to say, what are the 3 parts of the nucleotides? And one of the options is nitrogenous acid, right? Because I'm not going to try to trick you like that. In a way, if you want to shorthand it, you can just remember the 3 parts of the nucleotide are the base, the sugar, and the phosphate. Now again, technically it's a nitrogenous base, but technically it's a phosphate group, but I'm fine with the shorthand. If you can memorize the 3 parts of the nucleotide are the base, the sugar, and the phosphate, then you're good. I'm not going to try to trick you on the multiple choice options. And again, before we move forward, unless you forget it, the most important thing I think you should need to know, remember I gave you all the details about these three, the most important thing you should know is that it's nitrogenous base that defines which nucleotide it is, because that's the only part that varies. Which is what we're about to talk about, excuse me, on the next slide. Does anybody have any questions about this slide? All right, so again, there are 4 different nucleotides for our DNA. Same with RNA too, but on this slide, we're going to talk about the DNA ones. Actually, yeah. Now, of those 4 types, the A's, the T's, the C's, and the G's, there's actually, those are subcategorized, right? So technically, thymine and cytosine are single ring structures, as you can see here. Thymine and cytosine, those are single ring structures. And then the A's and the G's, the adenine and the guanine, those are larger double ring structures. Guess what? You don't need to know any of this. That whole bullet point, I'm not going to ask you. You do need to know there's A's, T's, C's, and G's, but you don't need to know which one's single ring, which one's double ring. You don't need to know what we call the single ring or what we call the double ring. Now, the fact that one of them is single ring, or some are single and some are double, will be slightly important later. The memorizing which is not important. Not for 100 level, not a biology major class. Any questions about this slide? Okay. So the next word for attendance will be bases, or even base. So use that in a sentence if you're watching the video later on. So not if you're watching live stream right now, but if you're watching the video, send a sentence that uses the word base or bases. Like, my uncle was stationed at a base in Vietnam, or I stole second base last time I played baseball, or even though it's spelled different, I like to play the upright bass, whatever. So no questions about this? All right, here's some other stuff you already know, but we're saying it again, and you need to know this. RNA and DNA have the same chemical structure, but there are some exceptions. You need to know the exceptions. One of which I've already said all of this in previous chapters, but one of these things I've already said so far today, which is that RNA doesn't have a T, it has a U instead, right? So RNA does not have the thymine nucleotide, it has uracil. So T instead of a U. That will be important later. I mean, it's important now, I'm telling you, you need to know that. But later on too, you're gonna have to do something called transcription, where I'm gonna give you a code of DNA. And then when I give you that code of DNA, you're going to have to figure out what the code of RNA is. So whenever in your mind you think there needs to be a T, if it's RNA you're dealing with, then there shouldn't be a T. As a matter of fact, you can write that down as a hint for the exam. If there's any question on the exam that says, hey, here's a strand of DNA, I want you to build a strand of RNA, right? And it's multiple choice, right? So if any of the multiple choice options have a T in it, you know it's the wrong answer because RNA does not have a T in it. So that'll help you on the exam, help you eliminate some options, right? If anything's asking you about RNA, if any of the multiple choice options have a T in them, you know that is not the correct answer. Another thing that you do not need to know is this next bullet point, which is that it's different sugars, which is why they have different names. RNA has something called ribose. DNA has something called deoxyribose. That is slightly important for people who work on these things and actually, people who are doing research. But for you guys, it's really useless. In a sense, it's been probably the only important reason to even bring it up is just so you understand why they have different names. Like, why do they call one RNA and one DNA? And now you know. So any questions about that slide? If you download the PowerPoint, you can see this little video here, this little animation that talks about DNA and RNA structure. Of course, there's a lot of better videos out there, and I've actually already shared them with you. So if I were you, I would go to the list of videos for independent work and look at those before you look at this one. But another thing is there's this thing called the Hershey's Chase experiment. I'm not actually going to get into this with you. You can watch that little video if you want. As a matter of fact, for independent work, you can look any of this stuff up. All these things that I told you, like what I was talking about, started in the late 1800s this, in the 20s this, in the 30s this, in the 40s this, in the 50s. Right? I told you all the things that we learned. What I didn't tell you was all the experiments they did to figure those things out. Like that's something I didn't even learn until I was a senior in my undergrad. So you don't need to know it, but if you want to look it up for independent work, go ahead. Like, so basically what I'm saying is don't take my word for it for all these things. You can go see, go learn how they figured these things out. Speaking of which, let's talk about how they figured out that last thing that we talked about, which is the structure of DNA. Remember, like I keep saying, they knew all these things about DNA, but they didn't know how it worked. They knew what it did, but they didn't know how it did. Like, how does it store the genetic information? How does it copy the genetic information? How does it pass the genetic information on from one generation to the next? They knew they did all these things, but they didn't know how. So again, your old textbook kind of tells this as a story, so I am too. Excuse me. So let's jump into the story of how they figured it out. It's interesting that they put it like this. They said James Watson and Francis Crick determined that DNA is a double helix. That is true. You do need to know what double helix is. It's funny that that's how they would put it, was Watson and Crick that did it. Watson and Crick didn't get the credit for it, but there was other people involved, and we'll talk about that a little bit in this chapter. But you don't need to know who did it. This isn't a history class. But yeah, you need to know that double helix is a spiral with uniform diameter made of 2 polynucleotides. I think the more important thing here is that you need to know it's 2 polynucleotide strands. I mean, yeah, it's a spiral. Yeah, it has a universal diameter, whatever. The important thing here is, remember, like when we talked about a polynucleotide, right? It's the sugar phosphate backbone. It's a string of nucleotides put together. But technically, when you see that DNA picture, that double helix, what you're looking at is 2 polynucleotides put together. So any questions about what a double helix is? All right, so again, your old textbook kind of tells this as a story, so am I, but you do not need to know any of this information I'm about to give you is regarding the people or the process and how they figured this out. But yeah, how it all started was this guy named Watson, James Watson here. He saw an X-ray image of DNA that was produced by someone named Rosalind Franklin. And when he saw that, that kind of set off all these events. And between the two of them, Watson and Crick did some work based on what they saw from Franklin, and they basically figured out the structure of DNA. And it wasn't just Watson and Crick and Franklin also. There was other stuff. So again, remember I told you there were some things that we already knew, or they already knew about DNA. Like they knew this was bonded to this and that was bonded to that. They just didn't know the 3-dimensional structure, right? And again, the 3-dimensional structure was important. We'll talk about that in a second. But you can look them up if you want for independent work. These two are really interesting, in my opinion, because like they're some of the most famous people in biology, but they were not good students. They like to party, they like to have a beer. They were not studious, so it was just like good luck that they had to find what happened upon this. And yes, you can look up their story if you want. Actually, who's done the video for the lab? Yeah, did they talk about them a lot? Do you remember? No, that's the one about, it's the one about the origins of humans, right?
Yeah.
Yeah, never mind.
Yeah.
So there's another video. I'm gonna try to find this other video. It's actually a movie. It's not that great of a movie, but it does at least show how they weren't good students. But anyway, the model of DNA is like a twisted ladder, like I've already said. And again, that's not really important that you understand that it's twisted. But again, like I said, it is important that you know that we were dealing with 2 polynucleotides. And if we're talking about a ladder, in that analogy, the polynucleotides would be the sides of the ladder. And those nitrogenous bases that we talked about that define the nucleotides, those are in the middle. And that, again, that'll be important later. But for now, not too much, not that important. Here's a picture from your old textbook, because since they used the term twisted ladder, I guess they wanted to show what that would look like. So if you can imagine a rope ladder and you twisted it, then there you go, that's what it would look like. More importantly, here's some other pictures, but again, not from your textbook, this is from your own textbook. 3 different ways of looking at DNA, right? Here's one way, the ribbon method, or the ribbon model, shows the sugar and the phosphate, it shows the nitrogenous bases facing each other in the middle. Same thing here, we got the phosphate, sugar, phosphate, sugar, phosphate, sugar. We got the nitrogenous bases facing each other in the middle. Here's something important, and this will come back, I'm gonna bring it up later. Right now, I'm just previewing it. But I don't know if I taught you this or not, but you definitely know that a straight line is a covalent bond. I did teach you that. Remember, in lab, you had to draw molecules, and you had to draw lines in between the carbon and the other carbon, for example, right? And one line was a single bond, 2 lines was a double bond anyway. So all these lines, that's still the same. All the stuff that you drew, this is the same. When you see a line, that's a covalent bond. What I didn't teach you is when you see a dotted line like this, that's a hydrogen bond. And again, this is going to be really important here in a second. Because remember, like I said, covalent bonds, these bonds right here are the strongest bonds. The weakest bonds, and I told you this was important back when we were talking about chemistry, is the hydrogen bonds. So here we have really strong bonds. Here we have really weak bonds. So imagine this wasn't a picture. Imagine there was actually a model right there, and that was real. Imagine if I grabbed these, I don't know, by the phosphate groups, and I started pulling. And I pulled and pulled and pulled until something broke. What do you think would break first? You think it would be these hydrogen bonds, or do you think it would be these covalent bonds? I'm just pulling, trying to pull it apart. Which ones do you think would break first? Which bonds? The covalent bonds or the hydrogen bonds? Don't overthink it.
Hydrogen.
Good, yeah, hydrogen bonds. Because like I already told you, those are the weakest of the bonds. Covalent bonds are strong, hydrogen bonds are weak. So this thing is built to be pulled apart, and that's going to be important later in the chapter. You can pull it, and all you're going to do is separate this polynucleotide from that polynucleotide. You'll still have a fully intact polynucleotide here. You still have a fully intact polynucleotide here. They just won't be connected to each other anymore. That's all. And again, that'll be important as we move forward. Right now, that's just a preview. Excuse me. If you download the PowerPoint, you can watch this little animation about the DNA double helix. Back to the story. In 1953, Watson and Crick published their paper that proposed the molecular model for DNA. So they figured it out. In 1953, they published it. If you click that picture of the Nobel Prize, there's a short little video that talks about this. You can do that if you want. Again, you'd have to download the PowerPoint for that to happen. Then, in 1962, so nearly a decade later, Watson, Crick, and Wilkins, who I didn't even tell you about, he kind of helped a little bit too, they received the Nobel Prize for coming up with the molecular structure of DNA. So again, it seems trivial to us. To you, it's just some boring thing that you had to learn in chapter 3, and then I'm teaching you it again, right? It's like, oh, who cares what the structure of DNA is? But again, remember, we knew all these things about DNA and what it did. We just didn't know how it did it. So if we didn't know how it worked, like how it did those things, then that was a major roadblock into learning new things. So again, as boring as this might be to you, this was really important back in the day, hence the Nobel Prize. But again, I'm not going to ask you any questions about the history here. But are there any questions about that side? All right, in this 1953 paper, which you can see right there on the right, Watson and Crick wrote that the structure immediately suggests a possible copying mechanism for the genetic material. So it was like a eureka moment. It was groundbreaking. Once they figured it out, the thing I just showed you, they're like, oh, this all makes sense now. I get how it works. Now, they had to prove that with later experiments, but knowing that there was this strand that was covalently bonded to itself, and then this strand that was covalently bonded to itself, and those two strongly covalently bonded strands were weakly hydrogen bonded to each other, that thing I just showed you, they knew that immediately. It was like, oh my God. Obviously, that thing was meant to be pulled apart. Make sense? Anyway, so that's why it was so important. Any questions about that side? Speaking of that, now that we know that once they figured out that, yes, that thing was meant to be split apart, and they figured out then how DNA can be copied, how DNA can do this, that, and the other thing we talked about earlier. Let's actually talk about it. How does DNA replicate? As far as the exam is concerned, this is a more important bullet point than anything we've talked about so far. You need to understand DNA replication. Here we go. As you already know, when cells reproduce, they have to duplicate their information, right? So you already know this from the mitosis meiosis chapter, which will be included on this exam. So this is a good reminder, right? Remember, in S phase of interphase, those chromosomes need to duplicate themselves because eventually they've got to split themselves apart, right? So they have to double all their genetic information. And that's what we're going to talk about in this chapter right here. Right? Because again, those chromosomes, those sister chromatids get separated. And what you want is two new cells that have the same copies of the original DNA. So again, you already know what happens in the S phase of interphase, but now we're going to get into more detail, like what exactly does happen in the S phase of interphase. So let's jump into it. This is a very great basic picture, better than I was showing. Again, remember, this blue strand is covalently bonded to itself. This blue strand is covalently bonded to itself. These two blue strands are hydrogen bonded to each other. So we know it's just meant to be pulled apart. And what happens is it's pulled apart, and I'm going to talk about this in more detail later. Right now, I just wanted to give you the overview since we have this picture. It's pulled apart, and the code is red, right? So here's an A. So the machine that does this says, all right, if there's an A, I need to bring in a T. So eventually, I'm going to try to draw that. Eventually, a T is going to be brought in and added to the mix. All right, here's a C. So eventually, there's going to be a G brought in and added to the mix. And that's how it happens. There is a question on the study guide that will probably be on the exam, and it has something like, all right, after DNA replicates, what are you left with? And what you're left with is two double helixes, right? Here's one, and each one of these this double helix consists of one old strand and one new strand. That will be a test question most likely. It's definitely a question on the study guide. When DNA replication occurs, the new molecule, the new double helix, consists of one new strand and one old strand. And that sounds like a trivial question, but it's not. Because for you to memorize that, what you need to know, which is not trivial, is that the new strand, the old strand right here in blue, serves as the template for the new strand. So again, there's an A right here, so that serves as a template. So there's going to be brought in a T, right? A new T to help build this new strand. There's a C here on the old strand. So we know we need to bring in the G to be added to the new strand. So the old strand acts as a template for the new strand. The next word for attendance is old. Again, same stuff. If you're watching this video later, if you're not streaming it live right now or in person, instead of just sending me the word old, use it in a sentence. All right, if you download the PowerPoint, you can watch this video that shows DNA replication in action. It's really cool, I think. It's a great three-dimensional rendering. It'll show you the DNA being separated. It'll show you these nucleotides being added one by one, right? So that dark strand is the old one. This light, the light colored one is the new one that's bringing in new nucleotides. Look it up if you want. Then you can watch this one, which looks less cool because it's not three-dimensional, but it's easier to follow. And then you can watch this one, which is kind of a mix between the two. It is kind of three-dimensional, but it is also a little bit less complicated and less realistic than the first one. Therefore, it's a little bit easier to follow. Speaking of which, if you do happen to look at this, I'll go ahead and tell you there's some words you do not need to know, like leading strand, template strand, lagging strand, template strand, primase, helicase, there's a bunch of other stuff. So when you're studying this, study from the textbook, study from the PowerPoints, and if you study from any other source, which I always recommend anyway, it's good to see how this source describes the process and how that source describes the process. But if you do, make sure you refer back to your textbook, because if there's any words from any other source that don't make sense to you, just ignore them. For example, you do need to know this word, DNA polymerases. Now, in all actuality, I'm teaching DNA polymerase as if it's one molecule, one enzyme. But technically, in all actuality, it's a team of proteins. So again, if you were to look this information up somewhere else, you would be getting extra words possibly. So don't let it confuse you. But yes, what is polymerase? Like I told you way back when I taught you about enzymes, enzymes are named after what they do. Well, I said they ended the word ACE, and they ended ACE, and then they're generally named after their substrate or what they do. So this will be a test question. It should be easy. It's right there in the name. What do DNA polymerases do? They make DNA polymers. These are the things that copy the DNA. These are the things that create the new strand of DNA. Like I already told you, there's the old strand, which on this slide I'm calling it the template strand. I already showed you that picture. They're opened up, right? So the old strands serve as a template. The DNA polymerase comes in and reads it and says, all right, if here's a C on the old strand, I need to bring in a G to add to the new strand. There's an A on this old strand, I need to bring in a T to add to the new strand. So again, the important things you need to know on this slide are, what does this? What is the enzyme that does DNA replication? And it's called DNA polymerase. And again, the other important thing that you need to know for the exam is it's the old strand serves as the template on how to build the new strand. What you don't need to know, but it's interesting to know, is that it's fast and accurate. How fast? Well, it does 50 nucleotides per second. That's really fast. And how accurate is it? It only makes 1 in a billion mistakes. So after doing a billion nucleotides, it'll usually have 1 mistake. Also, it has ways of fixing that mistake. Also, DNA polymerase can also repair DNA. And that's where we'll end off. So remember, I will see you online for lab at 10 o'clock. We will not be meeting in person. And that's it. Yeah, no more words for the day.